That's it.
So good evening, everyone. Thank you for being here. So we are going to give the second Dr. Cayman Metronomic Global Health Initiative Award. And before giving the prize, we would like to tell you a bit the story about Metronomic Global Health Initiative and the story behind this prize, and so that you know a little bit more about Dr. Cayman, who is a very unique person and very important person in the field of metronomics. So to start with, we launched the Metronomic Global Health Initiative now 4 years ago, and the idea was to try to promote the use of metronomic chemotherapy in children living in low and middle-income countries. So we first just built it slowly, a network of people who were interested. So they could be scientists, they could be physicians, or they could just like people who were interested and wanted to support us. And then we built a website, and on this website we have like posts of new publication, important publication, meetings, and so on. So you can have a look if you want to, whenever. And then of course we try to do some work, and at the end of the day just have some work published. So far over the four last years we published around 20 publications, or contributed to 20 publications. We tried to help organizing meeting or be present at meeting. So over the last year, these are the main metronomic meeting: Haifa 4 years ago now, 2 years ago Milano, Sao Paulo 3 years ago, and now Mumbai. And we have to decide tonight where is going to be the next meeting. Very likely Belgium or the US, and or maybe Canada, but it doesn't seem to be moving on nicely.
January or February.
Right. So that'd be, yeah. That'd be metronomic in the snow. And then we try also to do some preclinical work, and we've been working recently a lot on drug repositioning, and more specifically on propranolol, and you've heard already in the meeting the work about angiosarcoma. And we try to help setting clinical trials. So we have some in France. We completed 2 in Mali. There's one that hopefully should be starting next year in Brazil. There was the angiosarcoma project in India, and there's one ongoing phase 2 trial in Morocco. And so this is an example of some pediatric experience that are going on in low and middle income countries. So we're not implicated in all these studies, but there are some stuff going on and more to come. So it's just to give you a small view about it. So why do we think that metronomic is potentially interesting for, well, children, but also adults living in low and middle income countries? First of all, there's clearly an unmet need. There are some patients that do not have access to treatment at all, and some for which like it's complicated to have access to treatment, and sometimes the cost of treatment is a big issue. Toxicity of treatment can be a major issue. Abandonment of treatment also occur very frequently and much more often than in high-income countries, and you cannot give some sophisticated treatment because of the lack of infrastructure. So basically, when you use metronomic, you manage to overcome like most of these hurdles, because it's easy to give, it has a low toxicity or lower toxicity, and the cost usually is very limited.
Okay, so yesterday someone asked me for how long have you been working in this field, and I explained that person that I considered myself, that's not the end of the slideshow, could you put back the slides on? It's black, but it's because there's an animation. Thanks. Yeah, I'm a child of metronomics. Basically, when I was an undergrad in 2002, the first papers I read was the paper that Bob mentioned this morning by Timothy Browder et al. Obviously the paper by General Clements and Bob Cabell and his team, and also this editorial by Dr. Cayman. And you probably noticed I'm quite young, and so I'm still, playful in my head. So to me, these guys are my heroes, so I always like to portray my heroes as, superheroes of some kind. So for me, Judah Folkman was kind of Gandalf. He has so much wisdom, and Timothy Browder could be Frodo. Basically, General Clement is my Princess Leia, and Bob Carable is Master Yoda, of course. So who else can he be? And in this story, we didn't talk a lot about Barton over the last couple of days, but for us, the two of us, is definitely Obi-Wan Kenobi. And so the reason he's Obi-Wan Kenobi is because he made some major contribution to this field, and especially this really important paper he wrote with Bob back in 2004. And on a personal note, the reason we have such high regards for Bart is because he's always been very supportive of the work we've been doing together. So to give you an example, this very first clinical study that we've done in Mali in collaboration with Fousseni Traoré in Mali, well, we published it in his journal, in Barton's journal, and Barton wrote an editorial with General Clement. And that was kind of the beginning of our relationship together. And so we wrote a few pieces together for the JCO and also a review. But most importantly, we exchanged a lot of ideas. So he was really always encouraging us to pursue ideas that we had, and he was a great mentor. So there are some people in this room that I truly admire. Bob is one of them, Yuval, Shripad, and they will be able to tell you that when I admire somebody, I never stop telling them. I send them love letters as emails long like this, and I drive them a little bit insane. Am I not, Bob?
Really long.
Really long emails. And so one day I wrote an email to Bart to tell him that he was my biggest idol, and his response was, "Are you really sure I'm your biggest idol? Because if you look at this picture, I'm not that big actually." He inspired us in a lot of ways, and he was very visionary on a lot of different topics. And Sebastian yesterday presented this review that we wrote together with Nicolas and our team. And really, he was the one behind this, because he kept telling us, you know, people think that mathematics is something of the new century and we need to bring mathematics now in oncology, whereas mathematics has always been there. And behind EMTD, dogma, and behind all of this, there's mathematical modeling. And Bart kept telling us this, and so when we wrote this piece, we wanted to dedicate it to him, and so that's what we wrote in the acknowledgement, to say that he was the one behind this. But then we thought, you know, who reads the acknowledgement in papers? That's maybe not good enough to honor his memory. So we decided to create this prize that we named the Barton Kamen and Metronomics Global Health Initiative Award. The idea is to recognize outstanding work of medical doctors and researchers in the field of metronomics, so either metronomic chemotherapy and or repurposing of drugs in pediatric oncology. And so it won't be a surprise to anyone why our inaugural recipient of this award was Dr. Shripad Banavali. We gave this award 2 years ago in Milan in recognition of his work, and once again we're really proud that you were the first recipient of this award. And so now we're going to give the second award, and we're really proud of this, and I'm going to let Nicolas announce the winner of the award.
So it's like the Oscar almost. And so the second Barton Kamen Prize is given to Giselle, so new for work in pediatric oncology. And so I don't think we have to justify the price, but I want to explain to you what we wanted her to receive it. The first thing is that she was working with Bart, right? And I remember, I didn't know her at the time, but I remember he mentioned he was working with that great pediatrician working on Nefertimox at the time, and the beginning of the personalized therapy program. And so I thought, well, it's kind of fair to just like recognize that they were working together, and that Bart loved Giselle. And the second thing, that the work she's developing, and she'll have a brief presentation right now.
Okay.
Well, yes. Putting you on the spot, I mean.
The slides are ready, if you didn't know about it.
Okay.
And she's working on, in personalized therapy, she's working a lot on drug repositioning and metronomic therapy to some extent. And some of her work can really be applied in low and middle-income countries, and she has already started thinking about it. So I guess giving her this prize will also keep the movement going on. So there it is. I know what the prize is.
Thank you. Oh my God, I'm so surprised.
So that's why we're going to use this thing.
Okay. Yeah.
We have to move to the center.
Tu give it to her? Yeah. So she's out, huh?
No, it's on me.
Oh, it's on it is? So just don't do like Bob, who just like fed this morning and without giving a word. And so we expect a little talk and then the real talk.
Okay, wow.
Congratulations.
Yeah? Okay, well, thank you. I'm very surprised. But thank you so much for, this award. This is a huge honor to me, and I can't tell you how much it means. Dr. Kamen was one of my very first mentors, and he really inspired me to continue this work, to begin this work. Our first clinical trial in molecularly guided therapy would not have happened without Dr. Kamen, and it's been a pleasure to work with all of you and to be here today, and I can't thank you enough. And I'd love to tell you a little bit about the work that I've done with Dr. Kamen. Should I?
Just like stick your finger in.
Maybe. Nope.
Well, you gotta keep talking about it.
Okay. So this is the newest version of the trials that we started with Dr. Kamen. When we first started looking at genomic sequencing of our patients' tumors, the very first trial we did with Dr. Kamen was really just doing RNA expression profiling, which is what we had available back then in a CLIA certified laboratory. And it was a small study of 12 patients as a feasibility trial to see could we molecularly profile our pediatric cancers, at that time incurable patients with neuroblastoma who had multiply relapsed, and see if we could identify targets and find therapies to provide them in a metronomic dosing and good quality of life to extend the lives of these patients. And Dr. Kamen was the safety officer for the trial, evaluating, as we were bringing drugs together that may not have been brought together before, safety was a big concern for our IRBs. This is novel and new and hadn't been done in pediatric cancer, and he inspired us to do this. Since then, we've had multiple iterations of the trial. And now we've moved from exomes to, then we added DNA mutation panels, and then went to DNA sequencing, and now added RNA sequencing as well as the DNA sequencing. And that's the current trial. This is funded by Dell Computers. In collaboration with TGen. Dr. Jeff Trent is also one of my mentors who does all the sequencing on this clinical trial, and his team of bioinformaticians and genomicists at TGen are very critical. So this is now beyond neuroblastoma, but for any incurable pediatric cancer. So when a patient has passed upfront chemotherapy, then we can do a biopsy of a patient's tumor. The tumor is sent immediately overnight to TGen. Within less than 2 weeks now, we're able to have the exomes and RNA-seq data back. That goes through our computer program where we've created rules looking at over 200 FDA-approved drugs that have pediatric dosing, not necessarily chemotherapeutics, anything that has a target. And from that, a report is generated for us to look at. And then the tumor board, molecular tumor board meets, which I'll talk about in a moment, online. It's a virtual tumor board. This is now at 20 hospitals across the U.S., as well as with Raya Saab in Lebanon and with Nicolas soon in Marseille as well. So what's nice is that while there's 200 drugs that are possible, it can be applied in any country, because during the tumor board, we only focus on the drugs. For Lebanon has their own list, only the drugs that are available there for those patients. And then the molecular tumor board will create an individualized treatment plan with up to 4 different drugs combined together to treat the patient, and then we follow that. In addition, the tumor samples are also sent to our laboratory where we grow the tumor cells in culture and create xenograft mice models of each patient for future research. And that also becomes a resource then for future fellow, anyone, collaborator projects. This is just exact explanation of the DNA sequencing and the RNA-seq that we look at. And we look at not only mutations that are there, but translocations, inversions, amplifications, and for the RNA sequencing, we look at expression of genes and pathways. So far, this is a little bit old. Our initial study was the 14 neuroblastoma patients. Then we went to a second study where we did the 48 patients of a mixture of tumors. And then our current study, we're actually at 60 enrolled to date. So we have over 100 different pediatric cancers available. And then this is the molecular tumor board. What's been really interesting in the molecular tumor board is that clinical decisions aren't only being made by oncologists anymore. The pharmacists play a key role in helping us with dosing and putting drugs together that haven't been put together before, looking at side effects, toxicity profiles, and really making sure that this is safe for the patients. And the bioinformaticians and genomicists, one of the lead bioinformatics doctors from TGen spends a significant portion explaining to us the genetics of the patient's tumors and what they mean. Are the mutations activating or not? Or are they turning off? And so it's really important for us to learn from them in how to treat our patients. So it's been a great collaboration, and then the medical monitor reviews it. On our first study, that was Dr. Kamen, and then we go forward. But the predictions, our treatment of our patients are only good as our predictions. And so this is something that we work on in the background while we're treating our patients. So we're looking at bioinformatic correlation of drug response through high throughput screening with our biomarkers. And so to evaluate each of our rules. So as the patient's cells are growing in culture, we can then do high throughput drug screening. We use the Prestwick and NCI libraries and look at the IC50s and which cells are sensitive or not. And then we go back to the genomics. We see that these are 6 drugs that we're very interested in. We can see, like for this patient, MGT-0102, the bortezomib is much more sensitive than any of the other drugs, whereas a different patient, both neuroblastoma, the dasatinib is much more sensitive. So we see a variation in cell culture. Now if we want to correlate that with expression, we can see that each of those dots is a different patient. Some patients are sensitive, some patients are not sensitive to the dasatinib. The bottom axis is the expression level of the gene, and we can see as we go up in expression, and as we go up in, so this is the negative log of the IC50, so the higher numbers are actually the more sensitive ones versus lower for IC50. And so the higher expression of PDHFR beta, which is one of our rules for dasatinib. We have many rules, but this is the most predictive for patients for the dasatinib. And so we're going through each one of the rules, trying to improve those. And then we've also learned that the patients can cluster in different groups. So we're starting to incorporate in the computer profiling now, computer program, a patients like me module, where we can look at the genomics of a patient's tumor and see if we can find other patients that had similar genomics. What were they treated with? What were their outcomes? And learn from our previous patients now that the database is growing. From this study and looking at the genomics of our tumors, we've also transitioned into repurposing of old drugs. One of the drugs that kept coming up is a drug called tolcapone. This is a drug for Parkinson's disease, so not something that we would think about using in pediatric cancer. But the target for this drug is COMT. We found it overexpressed in many of our patients' tumor, and it would come up on their reports. It's just a little bit about COMT. Is important in the degradation of dopamine. As tolcapone blocks COMT, dopamine goes up, which is what we would want for Parkinson's disease. But this causes high dopamine levels in neuroblastoma cells specifically, is cytotoxic. It causes an oxidative stress and apoptosis. So we went to the laboratory, we got some tolcapone into our lab, and we looked at both patient cell lines and then established neuroblastoma cell lines. Oh, I gotta go. and found that it was sensitive in cell lines. It induced cleaved caspase-3 and PARP-induced apoptosis. And then in our mice models, we saw that tocopone alone was able to inhibit neuroblastoma growth. This is now a phase 1 trial. We've just opened, and in January, the first patient that went on had 5. He just told me this last night, so I would have had the slides for you. But there was a patient with relapsed neuroblastoma with 5 brain metastases who's been on the tocopone, and was on tocopone first alone, and then is now in combination with once a month oxaliplatin. But even with tocopone alone, we found that the tumors had hemorrhage and necrosis within the tumors, and the tumors became PET negative as well as MIBG negative, but they didn't change in size, which I thought was the talk yesterday was very interesting on how radiology can be different in terms of mechanism. So this patient is doing well. This patient's been on since the beginning of January with just this, and was a patient who had been referred to hospice. And so I think that this has been really helpful to learn. We're learning more, and we're looking forward to enrolling more patients on this trial. But and all our funding is primarily from either the Dell or parent foundations that fund pediatric cancer. Alrighty, thank you.